From the Netherlands, this is the Green Planet Monitor, voices and stories from a warm, wet planet. Today, powerful new antibiotics discovered using artificial intelligence.
We went into the laboratory, screened 7,500 chemicals against Acinetobacter. So basically what it looked like, instead of having to spend 3 weeks, let's say, or a month in the laboratory testing those 7,000 chemicals, we could train a model, push go to lunch, and come back, and then we had our predictions for us.
Sitting in a restaurant, staring at a piece of fish. Is it really that expensive kind? Pull out your DNA barcode reader and find out.
Well, that's the dream, having a Star Trek tricorder for sure. I could very well imagine this kind of device as a plug-in or an add-on to a smartphone. It does the laboratory part, but the computer in the phone does the rest. Connect to a database like ours, get the species name back, and display it on the phone.
And sex and the brain. Women's and men's are wired differently. Men's brains are sexualized before they're born.
The male brain has undergone a sexual differentiation, which the female brain does not undergo to the same extent in utero. So more chemical physiological changes have happened, and if something should go wrong, it could affect the male brain more than the female brain.
This is the Green Planet Monitor. I'm David Kattenberg. Before launching in, a request. Help us deliver this weekly package of Earth news. Subscribe and/or donate at greenplanetmonitor.net. It's a staggering statistic. COVID has killed almost 7 million worldwide. The number is likely higher. Meanwhile, a potentially deadlier pandemic sweeps the planet. Bacterial infections antibiotics can't cure. According to one UK government study, by 2050, 10 million may die every year infected by multi- or pan-resistant microbes that no drug will kill. Drug resistance is the inevitable result of antibiotic overuse or misuse. To treat viral infections, for example, what doesn't kill infectious bacteria makes them stronger. So, science searches for new drugs that do work. The trouble is, Big Pharma is not interested. There's no profit to be made developing drugs that will end up on the top shelf, reserved for emergencies when no other drug will work. Big Pharma wants to produce drugs that billions will use all the time. Precisely how antibiotics can't be used, or else they'd become useless. So, academic researchers are picking up the slack. At McMaster University in Hamilton, Ontario, a team of scientists are screening thousands of hopeful candidates, totally new kinds of antibiotics that bugs have never seen and are unlikely to become resistant to. And for hot tips, they're turning to artificial intelligence, neural networks, and machine learning. John Stokes is an assistant professor in the Department of Biochemistry and Biomedical Sciences at McMaster University. What is it that has led researchers such as yourself to artificial intelligence and deep learning and machine learning and discovering new drugs?
Yeah, it's a really good question, and one that we quite frankly think about all the time too. When we embark on any antibiotic discovery project, we are what I'll call tool agnostic. Our end goal is to find new medicines to make sick people not sick. Whatever set of tools will get us there most efficiently is what we're going to use. In the context of AI, we consider these algorithms as suggestion generation boxes. So in a conventional antibiotic discovery, a wet lab scientist goes into the laboratory and they might be able to screen 1 million molecules for some new antimicrobial chemical matter, let's say. But you can imagine, if let's say I want to kill Klebsiella, I have 1 million chemical library, I have to do 1 million experiments, right, to find the molecule that might do the thing I want it to do. There's a ton of laboratory automation that goes into this, but it's still, if I have 1 million chemicals, I have to run 1 million individual little experiments, right? And that's super time consuming and expensive too, because consumables, the dishes, for example, in which we grow these bacteria are not cheap. The acquisition of the chemicals to screen is not cheap. And what we often find too, in a lot of these chemical libraries that we can purchase and assemble in the laboratory, the chemical diversity that's often explored is not as vast as one would hope. So the question becomes, how can we improve upon this conventional laboratory screening process? And, I'm a proponent of the use of ML to help us, increase the rate at which we can discover new antibiotics.
Machine learning.
Yeah, machine learning. So the first thing that we have to do to train an ML model, machine learning model, is gather training data. We'll go into the laboratory and screen, let's say, maybe 10,000 chemicals against a bacterium of interest. So we'll go into the laboratory, collect a diverse collection of about 10,000 chemicals, test all 10,000 of those, and then we can identify which chemicals are antibacterial and which ones are not. Because we know the structures of all the chemicals, as well as their associated antibioticness, if we want to call it that, we can now train a machine learning model to understand chemical features associated with antibioticness and chemical features that are not associated with this antibacterial activity. And then once we have this trained model, we can then start showing it millions, tens of millions, hundreds of millions, billions even, of brand new chemicals in silico in the computer. And what it's going to do is tell us whether it thinks each chemical might be antibacterial or not based on its chemical structure.
And when you talk about the chemical structure, you're talking about its three-dimensional shape.
Yeah, so we're talking about atomic interconnectivity. So what the machine is learning, what the model is learning, are how all the atoms are connected to every other atom in a molecule, and how the atomic interconnectivity of every molecule is associated with its antibacterial activity. And it's learning, with the types of models that we often employ, often very subtle features that would be quite tricky for us as humans to pick up by eye.
In other words, whether or not there's a kink in the molecule somewhere, or it's got a curious spot on its surface area.
It could be one or more of many different types of features. What's kind of interesting about all this is we actually don't know what they're seeing. You'll often hear deep learning. That's machine learning using neural networks as the model architecture. That's called deep learning. So you'll often hear deep learning models colloquially called black boxes, because we feed input data. It does a bunch of stuff. It extracts features from that data and then gives us an output. But what it's extracting, we actually don't know.
And what's a neural network?
So a neural network is a type of artificial intelligence algorithm that takes inspiration from neuronal interconnectivity of the brain. You have an input layer that takes a whole bunch of data, and then a whole bunch of these things called hidden layers that are responsible for processing the input data and extracting relevant features. And then you have an output layer that is what the human then interprets. So the output layer might be quantifying antibioticness on a scale from 1 to 0.
And so you feed in 10,000 structures, which you've analyzed empirically in the lab.
Yep.
Using automation. And you say, these work, these don't work, these are kind of about halfway. And you feed that in, and the neural network comes back at you with what, like 200 best shots?
So after we've trained our model, let's say we train our model on 10,000 chemicals. So what that's going to do during the training process is the model is learning all of the chemical features associated with antibioticness, and then all the features that are not. So then let's say that's 10,000 chemicals worth of training data. Well, now we can show it, hundreds of millions quite easily of brand new chemicals it has never seen before. And then based on what it learned during training, it's going to take that input structure, that new structure of a chemical that it's never seen before, extract the relevant features from it that it learned during training, and then spit out a number between 0 and 1.
And some of these chemical structures, molecules that you're feeding in, are already pharmaceuticals that are used to treat whatever, hair loss, for example, or urinary dysfunction.
Yeah, they can be any molecule. They can be preclinical, clinical stuff no one's ever looked at before. It can be any chemical. And then the model, based on what it learned during training, will tell us whether it thinks it's antibacterial or not. And what's really cool, these neural network architectures, like I just described, are becoming sufficiently powerful, sufficiently sophisticated to extract a lot of really complex features from chemicals in a way that I think can meaningfully influence how we make empirical decisions, laboratory decisions. So going back to my suggestion generation box, let's say we run predictions using our trained model on 100 million chemicals. 100 million experiments in the laboratory is infeasible. It would take probably a decade to do that many wet lab experiments, testing 100 million molecules. But we can run predictions on 100 million molecules in a few days. Fine. And what it's going to do is it'll allow us to look at all the chemicals that it thinks could be antibacterial. So it's making a whole bunch of suggestions for us, right? It's saying, here's 100 million chemicals. I only think you should really pay attention to these 1,000, right? So it's making 1,000 suggestions for us. Then we, as the wet lab scientist, the person who's going to eventually do the experiment, can then look at those 1,000 suggestions, strongly predicted antibiotics, and then say, okay, which one of these 1,000 or which dozen, whatever, of these 1,000 am I going to test in the laboratory?
And the interesting thing that listeners should know is that antibiotics today, and for the last, since antibiotics were discovered 100 years ago, the first one, all of them, they all belong to maybe 5 or 6 different classes of structure. There are 5 or 6 types of molecules. Like, the penicillins and the cephalosporins are beta-lactam drugs, and the folate inhibitors have this shape. There's only about a half a dozen classes. So this process you're describing is going to be spinning off potential drugs that belong to totally different structural classes that bear no resemblance to any of the known structural classes of antibiotics.
Precisely. And the important, I'll say, machine learning feature in order to get us to these new structural and therefore functional classes of antibiotics is the ability of the model to, quote, generalize. So what that means is we're training the model on one set of chemicals, that live in a defined chemical space, let's say. But when we're looking for structurally and functionally new molecules, we tell that model, okay, we want you to look in some brand new chemical space for antibiotic, for antimicrobial chemical matter, right? And that process of generalizing to brand new chemicals that are divergent from those that were used during training is something, that has relatively recently been robust. And I'd say that we're within the last 5-6 years, something like that.
Tell me about Acinetobacter baumannii and why you chose it as a target for developing a group of potential antibiotics to treat it. Like, why Acinetobacter baumannii?
So I've had, I'll say, a casual interest in Acinetobacter for a long time now, like maybe 10 years, like since my mid-grad school days. And I find it really fascinating for a number of reasons. First, it's a hospital-acquired infection. It's a hospital-acquired pathogen. And that's where you find Acinetobacter baumannii, is in hospital settings. You don't find it out in nature very much. Now, the problematic features of Acinetobacter is that it's hard to remove from surfaces. You'll find it on doorknobs and hospital equipment. It's really hard to strip. And the other interesting feature of Acinetobacter that makes it a problematic pathogen is its ability to uptake and just kind of hold on to extracellular DNA. And you'd imagine if you're a bacterium living in a hospital, a lot of that extracellular DNA is going to encode antibiotic resistance genes. So it's really common to find multi-drug resistant, extensively drug resistant, and pan-drug resistant isolates of Acinetobacter.
These are superbugs.
Yeah, perhaps the superest of superbugs.
And they have these pumps. They have multifunctional pumps that pump out this antibiotic and that antibiotic and the other antibiotic. Tell me about that.
Antibiotic efflux, that's the process of pumping out molecules, is common amongst, clinical pathogens. It's exactly what it sounds like. An antibiotic molecule will make it into the cell, and then bacteria that are expressing these efflux pumps will just take that molecule and pump it right back out. So you actually never get enough antibiotic molecule into the cell for it to do the thing to kill the bacteria.
And Acinetobacter baumannii is really good at this.
Yeah, I'd say it's really good at every type of resistance, whether we're talking about efflux or enzymatic modification of the antibiotic, enzymatic modification of the target to which the antibiotic normally binds. There are many mechanisms through which resistance can emerge, and I don't want to speak too generally, but I would bet that Acinetobacter does all of it.
And what kind of infections does Acinetobacter cause? And do people die of these?
Oh, yeah. So it causes a diverse array of infections too. It's quite virulent. So you can get things like pneumonia and meningitis, bacteremia, so blood infections, wound infections. And, estimates put mortality at 1 in 4.
1 in 4 people who come down in the hospital with Acinetobacter infections die.
About that, yeah. Yeah. It's like no joke.
And how are they treated? What antibiotics are used to treat Acinetobacter?
I can't recall first-line treatments, but I know for like drug-resistant strains, colistin is one that they'll often use. And colistin is it's a useful antibiotic, but it comes with a whole host of side effects. A big one being nephrotoxicity.
The toxicity to the kidneys.
Yep.
And colistin is the drug of last resort.
It's the last resort antibiotic, yeah.
And are there colistin-resistant strains of Acinetobacter floating around?
Oh, sure.
That's the scary stuff.
Yeah, there are isolates of Acinetobacter that are resistant to every clinically used antibiotic we currently have. And over time, the number of clinical isolates of Acinetobacter that are resistant to every antibiotic we have is just going to grow.
Tell me about a Bowson.
So we set out to leverage machine learning to discover new antibiotics against Acinetobacter. And as we were discussing earlier, one of the criteria for when we look for new molecules is that they have to be structurally divergent from any other clinical antibiotic we have. And that's for an obvious reason, and it's to avoid, being able to overcome, let's say, pre-existing resistance determinants. So how we went about discovering this molecule is we went into the laboratory, screened 7,500 chemicals against Acinetobacter, just looking for which ones could inhibit growth, which ones wouldn't. We trained a model on those chemicals, and then we started to run predictions on a relatively modest in silico chemical collection. So we only ran predictions on something like 7,000 chemicals in the computer. And we could have probably screened those 7,000 molecules in the laboratory. It would have taken about 3 weeks. Maybe a month, let's say. But for perspective, our model was able to assess those 7,000 chemical structures in about an hour and a half, 2 hours. So basically what it looked like, instead of having to spend 3 weeks, let's say, or a month in the laboratory testing those 7,000 chemicals, we could train a model, push go to lunch, and come back, and then we had our predictions for us.
And how big is the computer that is used to do this?
So we have access at McMaster. Our lab is super fortunate. We have access to a GPU cluster, graphics processing unit cluster, that we often do all of our neural network training and predictions on. But with a model this size, the one that we used to discover that narrow spectrum Acinetobacter molecule, that could be done on a laptop.
It could be done on a laptop. It's amazing. Yeah. So tell me about a Bowson. What is this molecule? It's a narrow spectrum drug. Talk about this. It's important. Like narrow spectrum drugs are like precision guided bullets that are less likely to generate resistance. So tell me about a Bowson.
So when we came across this molecule, we tested it in the laboratory. The model said test this, so we did. And we observed that it had, a reasonable, I would say actually a pretty good MIC, minimum inhibitory concentration. So that's the lowest concentration of drug that it takes to inhibit the growth of a bacterium in a dish. About 2 micrograms per milliliter. And that puts it roughly in the ballpark of other clinical antibiotics for Acinetobacter. So we were like, that's great. Let's continue studying this. And one of the experiments we often do is just test the spectrum in which the molecule could work. So we take a whole bunch of different clinical isolates from a bunch of different species and say, what is the spectrum of activity? And most antibiotics are what are known as broad spectrum, meaning that they inhibit the growth of a really wide phylogenetic spectrum of bacterial species. When we tested this compound, Abicin, against a whole bunch of different bacterial species, we observed it didn't work. It only worked against isolates of Acinetobacter. And even at really high concentrations, it wasn't working against, Enterobacteriaceae or Pseudomonas aeruginosa or Staphylococcus aureus. So when we observed this, we actually we just decided to focus on this molecule. We could have very easily continued to run predictions, right? That we were expecting to have to run a lot of predictions. But when we came across this molecule that was potent and had narrow spectrum Acinetobacter activity, we decided to put all of our effort into this chemical because it was just such an odd, it's a unique phenotype. Narrow spectrum activity is not common. So when we saw this, we knew we had something cool.
And the best drugs are narrow spectrum, all other things being equal, because they're only targeting one thing, and so if that thing develops resistance, it does. But other things that you're not even targeting, they don't develop resistance because you're not targeting them.
Exactly. So broad spectrum antibiotics impose a universal selective pressure across, an entire ecological niche. So it promotes the horizontal dissemination of antibiotic resistance genes, like really rapidly. But if you have a targeted narrow spectrum antibiotic, you no longer are imposing a universal selective pressure. You're only targeting that one nasty thing. So theoretically, the dissemination of resistance will be a lot slower.
So, I mean, the big question is, where does one go from here? When is this thing going to be put into clinical trials to treat Acinetobacter infections, and when's it going to become clinically available?
For preclinical, it's never seen use in patients, so it's still a long road to before we have this molecule, positioned for, let's say, even early clinical trials. But we are aggressively pursuing this compound through medicinal chemistry right now. So there are some features of the molecule that we don't love in the context of turning an antibacterial chemical into a clinical antibiotic. So we are structurally modifying the chemical in order to optimize some of the medicinal properties that we want.
And to reduce the side effects. I mean, the question is, I guess, does it have nasty side effects?
Yeah, so right now we're modifying the chemical to improve its solubility, improve its toxicity profile, improve some of its other, adsorption, I'll say anatomic localization properties. So we can ensure that the molecule gets to the right site of infection at a sufficiently high concentration for a sufficient amount of time to clear the infection. Because, we often think of drug discovery as I'm trying to find a drug that can cure X. And that's one property required of a molecule, but there are many properties required to make like a lead molecule into a clinical drug, because it has to be super well tolerated, and it has to have good pharmacokinetic and pharmacodynamic properties, and it has to do the biological thing you want it to do, and right?
So machine learning, artificial intelligence, deep learning, right? I mean, artificial intelligence gets talked about a lot in the mainstream media, and we hear stories that are scary about how artificial intelligence may be used to replace human beings, and it could pose an existential threat to humanity if improperly used. It's been in the news recently. But here we have an example of artificial intelligence and machine learning being used to do something incredibly productive and useful in medicine.
Yeah, and I'm not, what do you want to call it, an artificial intelligence philosopher, but I feel like it's very clearly a transformative technology that's going to have major implications in, I'll say, like professional and social arenas. It's gonna change a lot how we work, how we interact with others, etc. And just like every other transformative technology, I feel like it can be used for beneficial things and pretty bad stuff.
So last question, you've discovered a bousin. A bousin is an exciting new discovery. What other potentially powerful antibiotics has your laptop spun off and suggested to you should try this out? How many of them are there?
I mean, we're running predictions for new antibiotics against different bacterial species all the time. And I'd say at any given time, we have more than a dozen molecules that we're actively looking at in the laboratory to see which ones could be useful and which ones are unlikely to be useful.
More than a dozen.
About that, yeah. And that up and down a little bit. But when appropriately trained and appropriately leveraged, these algorithms are, like I said, they're excellent suggestion generation boxes. And they don't only work for bacteria. I mean, we have an amazing collaboration with a great friend of mine at the University of Guelph, where we've trained an artificial intelligence model to predict new antifungal compounds. That's a project that we recently started a couple months ago, maybe, and we've already had our first glimmer of success in that project.
Because fungal infections are notoriously intractable.
So I'll give you a sneak peek. The early data that we've acquired so far suggests that we have 2 molecules that are modestly potent, that are structurally divergent from every other known antifungal compound, that work at least in vitro in a dish against Candida. That one's still early. There's a lot of improvement, but the fact that we were able to leverage an artificial intelligence model that's quite similar to the one that we use in bacteria and see, these early signs of potential success in fungal species is super promising. And again, I think that this is going to allow us it gives us confidence to start looking for molecules that do, other biological things in other biological systems. So it's, we might do antibacterial work, antifungal work. This will probably work for antivirals and as well as human diseases, right? So the question is not necessarily, will this type of model work for disease X? The question becomes, how do I gather appropriate training data to train a model to apply it to disease X?
John Stokes is an assistant professor in the Department of Biochemistry and Biomedical Sciences at McMaster University in Hamilton, Ontario. Read more about his research at greenplanetmonitor.net. You are listening to the Green Planet Monitor. If you're appreciating what you're hearing, go to greenplanetmonitor.net and subscribe or donate or both. With your generous help, we'll keep delivering stories about global ecology, human development, justice, and science to listen to, read, and watch. Since ancient times, nothing has fascinated human beings more than the diversity of life around them. Aristotle was among the first natural historians. Naturalis Historia by Pliny is the Roman Empire's largest surviving work. The identification and description of living things has come a long way since ancient Greece and Rome. Today, biologists use DNA barcodes to identify living creatures. Here's a story about that. I'm sitting in a booth in a crowded sushi restaurant here in the Netherlands. 25 euros for a small slice of red snapper. That's 34 Canadian dollars. I wonder if it's the real thing or just a cheap substitute.
It's always around 1/3 was mislabeled, and it's not random, it's not an accident. You often see the value differences.
Dirk Steinke knows all about food fraud and how to spot it. He works at the Center for Biodiversity Genomics in Guelph, Ontario, home to the world's largest database of DNA barcodes. Among the many practical uses of DNA barcoding, identifying fish species. Steinke tells me about one group he helped out.
They went to their favorite sushi stores in New York City and cut a little piece off of the fish they got served, and then they sent their samples to us, and we did DNA barcoding. And the message back was, yeah, in 40% of what you had there, the label was essentially wrong.
What is a DNA barcode?
Well, a DNA barcode is a small fragment of DNA, characteristic for a species. And it's slightly different in another species, and so on and so forth, which allows you later on, if you probably find a remaining part of something, find a butterfly wing, you could actually take the DNA out of that, and you look for this particular fragment we defined, go back to our database where we collected all this information, and try to match it.
A half dozen standardized barcodes exist for different groups of organisms. COX-1 is used to identify animals. Botanists use a chloroplast gene. The internal transcribed spacer, or ITS barcode, is used to identify fungi. Bacteria have their own barcodes too. Within any broad group, that standard barcode varies a little bit from species to species. A digital device reads it just like a supermarket scanner reads the barcode on a can of soup or a roll of toilet paper. Predictably, the DNA barcode readers are getting small.
So they're starting to build small suitcase-sized laboratories that people can use everywhere. They can even carry them in the field. Take your sample, throw it in there, and everything is done in that little box.
We've recently upgraded because the little device is now a big device.
Kevin Baintjes' barcode system isn't big at all. Baintjes is a barcoder at the Naturalis Biodiversity Center in the Dutch city of Leiden. He shows me his device, the size of a computer printer, with slots for 5 small barcode readers covered in nanopores.
There's a little opening here where you pipette the sample in, and then it's on this chip with all the nanopores in it. There's a motor protein attached to all of the sequences.
A motor protein.
A motor protein, and the motor protein binds to the pore itself and then makes sure that the DNA is translocated through the membrane, through that pore. When the strand of DNA passes through the pore, there are slight changes in the current, depending on which base, so the A, the C, the T, or the G, passes through the pore. And based on that electrical signal that comes out, we can sort of translate it into the DNA sequence.
Converted to different colors, those 4 letters in DNA's quaternary code look like lines on a commercial barcode. Barcode sequences get uploaded to a database that matches them with known species. Like a virus checker on your home computer, the DNA barcoder flashes species names on a laptop screen in real time. Kevin Baintjes and his colleagues have barcoded 5,000 species of animals, plants, and fungi, just a fraction of known life forms here in the Netherlands. Countless others are totally unknown. Their toughest challenge, analyzing thousands of barcodes in a drop of lake water or a pinch of soil. Meta-barcoding, this is called.
So all the fish that swim in the water, they leave trace DNA behind. We can just filter it out and then sequence it in bulk. So this is the meta-barcoding part. We get a list of species just from a water sample.
Lots of times, the barcodes that pop up don't match anything in any of the world's barcode libraries. Other times, matching barcode and species is easy.
Every now and then, a bird flies into a plane engine. It's not really recognizable as a bird anymore, but still they want to know what species of bird flew into the plane engine. We use whatever is left to get some DNA out and then sequence it and then match it to the reference database to find out what species it is.
DNA barcoding can also be used to spot toxic mold in grain shipments, insect pests in wooden shipping crates, invasive fish in lakes, and powdered elephant tusk in traditional medicines. Art museums can confirm whether their 500-year-old French cabinet really is made out of cherry. Natural resource managers can assess the diversity of life in bodies of water and the effectiveness of remediation. In the world of medicine, doctors use bacterial barcodes to study the integrity of our inner world, our microbiome.
We used to use the gram stain as our method of analysis, which is, a very tried and true old microbiologic tool, but obviously not a very sophisticated way of assessing a microbiome.
Deborah Money is a professor of obstetrics and gynecology at the University of British Columbia. She studies microbial communities in the female vagina before, during, and after pregnancy, using DNA barcodes unique to bacteria.
16S RNA was the original barcode that's been used by most folks, but I partnered with this group in the University of Saskatchewan, and they had started to use something called Chaperonin 60. And this is a different barcode, but a very helpful barcode for the vaginal microbiome that allows us to show the different lactobacilli that are present. It really matters which one it is, and it matters which subtype of Gardnerella vaginalis it is.
Armed with barcodes like Chaperonin 60, physicians may soon be able to diagnose infections on the spot, just like Dr. Leonard McCoy on the Starship Enterprise.
Well, that's the dream, having a Star Trek tricorder for sure, which just tells you what species it is by holding it in your hand.
Indeed, miniature barcode readers are now on the market. Dirk Steinke in Guelph.
I could very well imagine this kind of device as a plug-in or an add-on to a smartphone. It does the laboratory part, but the computer in the phone does the rest. Connect to a database like ours, get the species name back, and display it on the phone. So then you sit in the restaurant, you order your fish meal, you take a little sample, if it's really fast, you don't have to wait very long, and then you can probably call the waiter because you've discovered it's not the right species.
Okay, let's try this out. What the? Hey! Hello? Read more about DNA barcoding technology at greenplanetmonitor.net. While you're there, subscribe. For $5 or more, you'll be helping to make this news package happen. You are listening to the Green Planet Monitor, a listener-supported podcast. I'm David Kattenberg. Men are from Mars, women from Venus. John Gray famously wrote back in the 90s. Science says it's true. Sort of. Male and female brains are wired differently. Sandra Whittleson is a veteran brain researcher with a collection of over 100 brains she's been gathering for years. Albert Einstein's was the first. The differences between male and female gray matter, especially in the language and speech regions, interest Whittleson greatly. Whittleson's studies revolve around brain lateralization, anatomical and functional differences between people's right and left cerebral hemispheres just above the ears, and how male brains get sexualized. At the tender age of 5 weeks, male embryos get doused in testosterone, changing them and their brains forever.
This is very time-consuming work, as you can imagine, and there are many things that one could do at the histological level, and we started to do something, at the most simple level, was to look at the depth of the cortex. The cortex is the outer rind of the brain. So if you can think of the brain as, let's say, an orange, you've got the peel, and then you have the pulp inside.
And the cortex is the gray matter.
And the cortex is the gray matter, and it's composed of 6 layers. And then the fibers, the axons and the fibers that connect these neurons from one neuron to another, either a neighboring region or some far distant, those are the fibers, and that's the white matter that it goes deeper into the brain. So we started to look at the depth and then the number of cells, neuron cells, through the depth of the cortex and in each layer, and then you would be able to get the packing density. And we were particularly interested in whether the cells were packing more in one area than the other, because the more they are packed, it means the more cells there are in a unit volume. But then there would be less room for synapses, for the things that allow one neuron to connect and communicate with another. And of course, it's the connectivity that is crucial for thinking. I mean, many things are, but the connectivity is important. And so we started with 2 cases, and the results were extremely different. And we couldn't figure out why the results could be so different. And you see, if they were so different, then it meant that we would have had to have a larger sample because there was so much variation. If what you're looking at doesn't have too much variation, you don't have to sample as many cases. And this was very time-consuming work. And so I remember my colleagues saying, you've got to sort of go back and see, was the post-mortem period different? Was the age of the individual different, etc.? And we had chosen two people to be comparable. And there was only one variable on which they were different, and it was sex. One was a man and one was a woman. And this was about 10 years after my previous paper, in which I had shown that there was less lateralization of visuospatial functions in children, in girls, than in boys. But I wasn't thinking of the sex variable, and I wasn't really pursuing sex as a factor in my research. So we published that, saying that the cell packing density in the language regions, in the posterior language regions, is greater in women than in men.
Packing is greater in
women
than in men. Okay.
Right. So in other words, there were more neurons packed into a little area.
In the language area.
Yes. Once we realized that sex was the relevant variable, we went on and did the study with 5 men and 5 women, so we had 10 cases. The paper was published in 95, but we knew what we were finding, before 95. So I began to try to look at the effect of testosterone in these regions. And to do that, we studied people with atypical sexual orientation, with the assumption that wasn't proven, but with the assumption that maybe gay men would have a different testosterone level than heterosexual men. Not so much necessarily at the time, at maturity when we saw them, because testosterone has its effects, throughout life. But a major effect of testosterone is in utero. And that is when the brain undergoes sexual differentiation. The reproductive organs and the genitalia, the external genitalia, are different in men and women. They are, innate. These are determined by whether the developing fetus has 2 X chromosomes or 1 X and 1 Y. If you have a Y chromosome, there are genes on the Y chromosome that cause the primordial gonads to become testes.
Right, so this is the sex-determining region of the Y chromosome, which is responsible ultimately for the production of testosterone.
Well, women have testosterone too, but the point is women don't have testes. They have the adrenal gland and they have ovaries eventually, but it's the testes that produce a lot of testosterone. And it's the testosterone that cause the reproductive organs and the genitalia to be male. And to some degree, the female is the default anatomy. Sexual differentiation goes on in other organs of the body. We know that the male brain is larger than the female brain. The size of the hippocampus, which is important for memory, the size and the anatomy of the hypothalamus, which is important for basic drives. Even more importantly, and even in a more complex level, the actual pathways and networks. If you study this through functional MR imaging that tells you which parts of the brain are active, you see that the parts of the brain in the limbic system, which are in the center of the brain, have different pathways and are active differentially in the male and the female brain. The female brain has a cyclic, this monthly, the thing that allows, that causes a woman to menstruate, so that there is a physiological monthly change that is going on in the female brain that does not happen in the male brain. So one could go on and list literally hundreds of anatomical and chemical differences between the brains of men and women, and this would be true, in other animals as well. The connectivity in a male brain seems to be greater within a hemisphere than between, whereas in a woman's brain, the connectivity seems to be more intercorrelated and more involved across the 2 hemispheres than within the hemisphere. So that even though girls and boys learn to speak, it doesn't necessarily mean that the same substrate and the same physiological basis is present in both sexes, even though the final product may be the same, because there are many different ways of going from here to there and of doing things.
And in all of these instances, would you say that the pattern is that women's brains tend to be more bilateral, that they're using both sides to a greater degree together, whereas the men are more lateralized?
Right. You know, let me make another comment about lateralization. I think that lateralization has become a much finer concept, in that it's not like there is a spot for language, let's say for speech production and another area for speech reception, but that what these new techniques have been able to demonstrate is that there are networks, there are pathways that are underlying, the perceptions and the cognitions. And sometimes they involve both hemispheres, sometimes they are within a hemisphere, so that the localization is much more than an individual gyrus or area, but it's much more like the whole network.
It's more distributed.
Yes, exactly. So in language, one is thinking more of the distribution of the back and the front components connected by major fiber tracts, and even to some extent what is going across the two hemispheres, because there are aspects of language like the prosody, the emotional aspect of speech, which is processed more in the right than the left, but it is still connected with the analytic aspect, with the content part of language. So taking many of the studies, it appears that there is a more generalized interconnectivity in the female brain than in the male brain. Like the networks are more specific, are more independent from each other.
In males.
Yes. The male brain is a more specialized organ than the female brain. That doesn't mean that it's better or worse, but that it is more compartmentalized.
And so the networking that takes place is more limited to the location. It's less distributed.
There are more functional units that have some degree of independence
in men
than in women. And in fact, the clinical literature show when a man suffers damage in some part of the language network, his aphasia, his loss of language, tends to be greater than in a woman. And so the male brain has undergone a sexual differentiation, which the female brain does not undergo to the same extent in utero. So more chemical physiological changes have happened to produce a brain in an individual who is XY than in XX. And if something should go wrong in the timing, in the expression of some genes, or of the timing of the expression of some genes, or even some environmental insult that is happening in utero, it could affect the male brain more than the female brain. The male brain is more specialized, more modified, has more independent modules. In a way, I think it's more vulnerable, and I think it has the potential for less plasticity. Because if something is specialized for something, it may be more difficult for that to be taken over by another area, or for it to take over the functions of another area. So in other words, what I'm saying is that I think the female brain is less vulnerable and has the potential for more functional plasticity. And that fits with the clinical data.
Are men more vulnerable to things like cerebrovascular crises? Like, have studies like this been carried out that if there is, I'm thinking of a a stroke as
see,
just
but
as an example of damage that happens in the brain in a specific location.
Yes, that's exactly what I was saying. So that when there is brain damage, whether it's a stroke or it's a gunshot wound, that men become aphasic more frequently and more severely than women with comparable damage. And that's because they have a more specialized brain, a network that is more devoted or more independent, and there isn't maybe some other area that can pick it up, so that if you have a more bilateral or a more generalized or a less lateralized representation or substrate of a function, and if part of it is damaged, you've still got something else.
And that's the situation with women.
Yeah, yes. What I did want to say was that by trying to study gay men, we found and reported that gay men have a much higher incidence of being left-handed. And this has been replicated dozens of times. So this means that left-handedness, which is associated with an aspect of brain organization, one factor has to be genetic, innate, which has nothing to do, with the behavior of being gay or being straight. And yet there is a strong correlation. In the 90s, we argued that this was support for a biological, innate, genetic predisposition for sexual orientation, and that it wasn't by chance or by culture or by environment 100% that determines whether someone is going to be gay or straight. There's no question that environment, experiences, culture is going to affect complex behavior like sexual preference. But it's important to remember that there's also a genetic biological factor that is causing sexual orientation as well. And that's because the brain is sexually differentiated in utero, just like the reproductive organs are. The brain is set in its organizational pattern by the time you're born. The level of testosterone in a neonate and at birth is as high or even higher than one ever has, even than maybe at puberty or equal to puberty. So what is happening later on in life may not be the most relevant factor. It's what happened very early. So the fact that you have a different organization of aspects of the brain in gay men, which has nothing to do with the behavior of sexual orientation, suggests that there is a biological factor. And of course, other people have found anatomical differences in the brains of gay men. So this brings me back to the point that I was trying to make, that, and I think that is very important in today's world, that now people are paying a lot of attention to gender identity. And what we suggested, and we used the word mosaicism, this was with my colleague Cheryl McCormick, we suggested that something could go atypical in the development of the brain, and that these different aspects of sexual differentiation may not take place in a completely correlated way, where I think this work becomes very relevant to the whole concept of gender identity, because there is a lot of writing today that suggests that gender identity is malleable, like, the color of shirt you want to wear today, forgetting that gender identity may well have a genetic, innate factor that leads to it, just like the genitalia are determined by our sex hormones in utero. Our brain is sexually differentiated as well. And the brain then leads to certain aspects of our behavior, one of which is gender identity. Just like the muscles, there's the slow muscles and the fast muscles, and there's a different proportion of slow to fast in males and females. And it asks, and they are immutable. And just because you change your hormonal input later in life, it doesn't change the muscles that you have. So it raises very important questions. When someone changes their sex and wants to participate in competitive sports as an opposite sex, the sports world has not been looking to neuroscience to find out what muscles does this person really have. So these are very major questions. The National Institute of Health has recently, put out in their mission statement that now when drugs are going to be studied in different disorders, whether it's heart disease, cancer, etc., they're going to study men and women differently because more and more studies are showing that the same disease and the same medication has different results in each sex, in each biological sex. So then what happens for someone who is transgendered or even just chooses to say that I'm a different gender? Where do they fit into the NIH? What drug would they get? Who are they? And the point that I'm trying to make is, as important as socialization is, one mustn't forget that a lot of our behaviors are determined to some degree with predispositions by our genetic inheritance. And that includes not just language ability and visual spatial ability and and strength, muscle strength, etc., but it also includes sexual orientation, and it also includes gender identity.
And these things are laid down very early in the course of the development of the brain. It's laid down in utero.
That's right. And I mean, there are and there are cases where people try to change, for various reasons, the actual gender identity, the actual gender and the phenotype of the individual. But you can't change the organizational effect of testosterone that took place in utero.
Sandra Withelson is Professor Emeritus of Psychiatry and Behavioral Neurosciences at McMaster University in Hamilton, Ontario. Read more about Withelson's groundbreaking research and the sexual organ between your ears at greenplanetmonitor.net. That's it for today's GPM, a listener-supported podcast coming to you from Earth Chronicle Productions in the Netherlands. Listen, read, and watch more Earth stories at greenplanetmonitor.net. Find us on Apple Podcasts, Spotify, Stitcher, and YouTube, and our home base, Blueberry. We're also on Facebook. Wherever or however you're listening, reading, or watching, please help us make the GPM happen. Subscribe and or donate at greenplanetmonitor.net. Shout out to CFMU listeners in Hamilton, Ontario, and CKUW fans in Winnipeg. By the way, the guy behind this guitar is Dan Weisenberger. Thanks for your brilliant instrumental themes, Dan. I'm David Kattenberg. Over and out. For now.